Intestinal juice is having proteolytic enzymes. Can you name one of such proteolytic enzymes? Proteolytic means they will be hydrolyzing proteins. One name. This is not trypsin. Right. This is then trypsinogen. We will not get trypsin. If we get, then by this time it will destroy the pancreas. We have discussed. What is the function of trypsin?
It's activate other enzymes.
This is one. First, how it will be activated?
Enteropeptidase.
Peptidases. Where you will get this? What is the source of enteropeptidase? Means somewhere they should be produced, no? So what would be their source? It is intestinal mucosa, the mucosal cell facing the lumen. So there are villi. Within this villi there are a lot of columnar epithelial cells. They will be producing. They will be produced in the crypt of liver cones. There are some cells, they will be produced by this. So when this pancreatic juice enters into the lumen, immediately or just beforehand, this is already been in the lumen. These are secreted because the presence of food in the stomach, it will initiate the secretion of intestinal juice beforehand. To some extent, it will be liberated. So now this enterokinase is ready. Enterokinase, entero means intestine. So this kinase enzyme is present on the intestinal wall. So they will activate. Activation means this trypsinogen, these are made of amino acids only. So they are having some extra amino acid. 2, 3, 4 amino acids are extra. They will split these extra amino acids. So trypsin will be the outcome. Now it is activated. Now this trypsin will convert all other proenzymes, inactive enzymes. This is one function. One function of intestine, small intestine, already we discussed. What? See, activation. By how intestine showed its activation function? By liberating enterokinase. So it has got activation action also. Okay, so now it has performed this. Now what will be there? What will be the secretory function? Intestine has got some secretory function. How can you say that? Because crypt of liver cones, they will be secreting the juice, plus the apical branch border epithelium, thus this epithelial cell, they will also be secreting some fluid. So all this fluid together is succus entericus or intestinal juice. So they are secreting something, and this secretion contains so many things. So secretory function it is doing, intestine is doing. Moreover, it is contracting, relaxing, making some movement in the intestine. So this will help forward movement of the we will find there are peristaltic movement, we will find there is pendular movement, different kinds of movement we will see. Okay, so when we are fasting, no? Say you didn't take breakfast today, so now it is 4 o'clock. So what is there? What your stomach will do? You may find sound also. So it is because of the movement. So what are these different movements we'll study in our next class possibly. So it is one of the function. It is helping forward movement of the food, and it is helping mixing of the food, half digested food or partly digested food with the fluids, intestinal fluids. Not only intestinal fluids, pancreatic fluids are also coming. Juice from gallbladder is also coming. So many masala. So all this together has to be mixed. So we can see intestine is performing secretory function. Another is, this is the mechanical function. So you understood mechanical function, secretory function. Now this is also doing a digestive function. It is also performing the digestive function. Intestine. How? By liberating various digestive enzymes. And these enzymes are proteolytic enzymes, amylolytic enzymes, lipolytic enzymes. And see, non-enzyme substances are also there. Let's see one by one, what is their function? So with this, they will be performing the digestive function. Say, now we'll see how protein is digested. If we start from, if we say how protein is digested, so we have taken food, whether any protein is going to be digested here? No. Because salivary juice doesn't contain any proteolytic enzymes. So protein digestion will start where? In the stomach. You may drink milk, you may drink meat, fish, other proteins. So there, this protein, digestion of this protein starts in stomach. Before this, the proteins will be converted into acid metaprotein in presence of hydrochloric acid. Now, digestion of protein will be easier by the pepsinogen. But pepsinogen is not yet active. So it will be activated, this pepsinogen will be activated by hydrochloric acid. So now pepsin is activated. Pepsin is a endopeptidase. Is an endopeptidase. What does it mean? You are a peptide. You are in a peptide chain. You are amino acids in a peptide chain. Endo means inner. These are terminal peptide bonds. These are terminal peptide bonds. These are terminal peptide bonds. They are not interested on terminal peptide bonds because it comes under another department. It is under biochemistry. So my department is, I have been told, you just delink the middle interior peptide bonds. So they will break it. As a result, what I will get? A smaller peptides I will get. If I break it here, I will get a tripeptide. Tripeptide. 3 amino acids. Okay, that's all. Here, this is not my department. That this peptide bond is not my department. I can break this, I can break this, I can break this. So in this way, peptidases, trypsin, will produce so many smaller peptides. And it may produce tripeptides. These are the products. So peptidases, what they do? Peptidases, one of the example of peptidases is trypsin. One of the example of trypsin, these are endopeptidases. They will break the inner peptide bonds, hydrolysis. As a result, what I will get? Suppose this is a very big protein having 200 amino acids. Having 200 amino acids. Okay, so if it breaks the middle one, then what will it? Even 100. So these are proteoses. Earlier one was protein, now these are protein, smaller, relatively smaller. Now if I get further 100, if I make 50, 40, 60 like that, then these are peptones. So gradually it is becoming smaller. So in this way, ultimately I can get peptones, smaller peptides, smaller peptides, and some tripeptides. Okay, so these are the products I can have by the action of pepsin.
What is the proteose?
Proteose. Proteose means, suppose this is the original molecule of the protein, having lot of amino acids. All are linked. So this is the total protein. So it has been transformed into acid metaprotein, but not splitted anything. It has just changed the tertiary quaternary structure. But all are in peptide bonds linked together. It has just changed the, I think it is biochemistry will teach you very important, this one, the structure of protein, primary structure, secondary structure, quaternary structure. Okay, tertiary, quaternary, these structures are very important because the proteins, particularly enzymes, they are inactive. When they are active, they are deformed. Some orientation is changed. As a result, the active group will be coming out. Now the active group or the catalytic part who will break or make hydrolysis. So this will be exposed. Now this is ready to act. Before that, it was hidden. It was in inactive form. So this quaternary structure, even when we will be discussing transport of oxygen by hemoglobin, we will see, you have also seen in your book, oxygen dissociation curve. It is, question comes, why the curve is sigmoid shape? Initially, it has got poor affinity for oxygen. That's why it is oxygenation, saturation of hemoglobin is low. But suddenly, its affinity increased. Sharp increase in saturation. And thereafter, again, affinity is dropped. So again, this will be flattened. As a result, what I will get? An S-shaped curve. Now, why it is happening? Initially, the hemoglobin molecule every hemoglobin molecule have 4 heme moiety. Out of this, only 1 heme is exposed. in quaternary structure. And all other heme moieties, these heme moieties are having iron, and oxygen will bind with this iron. So unless this iron is exposed, come to the surface, how the oxygen can bind? Not possible. Only one out of the 4 heme moiety, one heme moiety is exposed. So this one only can bind. If oxygen is available, this one only can bind. But others are there, but they are hidden. So once one molecule binds with that, immediately what happens? It starts to deform. As a result, the hidden heme moieties come out. And now they are ready to bind with oxygen. So means here, very rapidly, all are available. So, there was one boat. Now you are having 3 more boats. So, number of more students can jump into the boat. It's like that. So, affinity for once oxygen became bound to the hemoglobin, first heme moiety, immediately what happens? The affinity for oxygen is increased. That's why suddenly there is rise in formation of oxyhemoglobin. So this change in shape is important. That is change in quaternary structure. Even we have seen in transport proteins, when they open, channels open, actually what is happening? There is change in their quaternary structure. It leads to opening of the channel. When a ligand, you know ligand? I already told. When a ligand has to show its activity, it must have a specific receptor on the cell membrane. So when a ligand binds, it wants to create some changes in the cell. So before it cannot enter. So what it will do, it will find with the receptor. When it became bound to this, immediately its conformation is changed. Because of this conformational change, the one enzyme is there inside. Say adenyl cyclase. So before it was hidden, now because of conformational change, now adenyl cyclase is activated. So its duty is to convert ATP into cyclic AMP. Now this cyclic AMP is the second messenger. Now this cyclic AMP will make a lot of change within the cell. And through this change, the ligand, what it wanted to do, that will be done by this cyclic AMP. So this is the way how the hormones show their action. So the second messenger will vary. Here it is cyclic AMP. Somewhere it may be calcium ion. Somewhere it may be PP3. Somewhere may be IP3. So it will vary. But for steroid hormones, they can penetrate. The steroid hormones are lipid soluble. So cell membrane is not a barrier. So it will enter. But they must also have a receptor. But receptor lies either within the cell, cytoplasm, or within the DNA, or within the nucleus, or within the genetic material. And when they bind with this, immediately transcription or some changes will be there. These changes may be positive, it may be negative, it may be stimulatory, it may be inhibitory. Our functions are balanced. Some will excite, stimulate. If it is too much, then other action will be interfering. Okay, stop it. bring it down. So that's why when we are very much excited, somebody comes, be cool down. Okay, but there is none to cool down me. So peptidases, example of peptidases are, all these are peptidases. Trypsin and chymotrypsin are not secreted by succus and entericus. These are present, it is present in pancreatic juice. We are not discussing pancreatic juice. In succus and entericus, we will only get only these three: aminopeptidase, dipeptidase, and tripeptidase. We have seen in pancreatic juice there is one carboxypeptidase. And where is that lady? She was having free carboxylic group. But amino groups are engaged. Carboxylic group engaged with her. Amino group engaged with this carboxylic group. Amino group engaged with this. So your one is amino group, free amino group. This is free. And this gentleman, aminopeptidase, will start breaking this from the free amino terminal. So, come out, she's freed, amino acid. Now this is carboxylic group, free, break it, come out. So in this way, amino acid will be freed. And the chain will be gradually shorter. This is the action of aminopeptidase. But this juice is not having carboxypeptidase. What's the need? Pancreas is there. It will be done. It is under the department of pancreas. Okay? So next enzyme is dipeptidase. It's very simple. It's very simple. So they are dipeptide. One amino acid, they are linked. So dipeptidase will break this peptide bond. So two amino acids will be formed.
Function same as aminopeptidase only.
Only the substrate is dipeptide, not polypeptide, not tripeptide. For tripeptidase, first it will break, now dipeptidase, you leave, and to break this there is dipeptidase. Now it is under your department. Amar কাজ আমি করে দিয়েছি, now you do it. Okay? So, in this way, proteolytic enzymes of succus and entericus digest the proteins. Now let us see. I have taken a protein. What will happen? Here nothing will happen. This protein will be entering the stomach. What will happen? It will face pepsin. As a result, what will be the product? Digestion. Will it be completely digested? Partly digested. What will be proteose? Peptone. Gradually it will be smaller. Peptones and smaller peptides. And to some extent, some tripeptides or dipeptides. That much. It will not free any amino acids. So that much will be done. Even after that, not all proteins has been converted into peptones or this. All native proteins are also there. Some part of the proteins are partially digested. Now, all the native protein means undigested. Still they are undigested. You have eaten went to restaurant, so you took a lot of chicken. So this will take time, no? This is not possible for all the proteins present in chicken to digest there. So part will be there and the rest part will be done in the intestine. Come here, then I will make you completely digested. So that much will be done in the stomach. If you take instead of meat, instead of chicken, if you take milk, what will be there?
Lactase will be there.
Huh?
Lactase will be there to digest lactose.
The proteins present in milk. Can you name it? Casein. Yeah, casein. So pepsin will first, actually caseinogen. The pepsin first, it will convert it into paracasein. And this paracasein immediately, a lot of calcium ion will be there. So immediately it will bind with calcium ion and it will form calcium paracaseinate. And these are nothing but not card. In Bengali we call it chana. So with this compound we make rasagulla, various type of sweets. Paneer, paneer. Cottage, yes, good. Okay, so that now after that it will be done. And this paracaseinate, calcium paracaseinate, will again the pepsin will act on it. We will produce peptone as usual, smaller peptides, tripeptides. This will be the product in stomach. Now the food, actually, what is the last part of the stomach? Last part of the stomach? Pyloric end. Antrum? Antrum? Pyloric antrum?
Pyloric antrum, তারপরে pyloric sphincter.
Okay. So pyloric sphincter means it is obstructing. If in the absence of this sphincter, what would have happened?
Direct entry of chyme.
And this chyme is having lot of hydrochloric acid. They would have destroyed the
Wall of
intestinal wall. So this wall will face consequence. What is the consequence? Ulceration of duodenum. So we don't want it. We want limited, controlled release of this kind, of this food bolus. So, okay, your first bolus come. It will allow. So it has entered into duodenum. It is acidic. Immediately the wall sensed it. Intestine will sense it. So it is having reflexes. It will release. It will ask to release bicarbonate ions from pancreas, from bile. So they will cause neutralization of these acids. So they are preventing the injury first. Then they are making the environment alkaline. Because all these enzymes, pancreatic enzyme, intestinal enzymes, they act in the alkaline medium. So they are making the medium alkaline. Clear? So now this way controlled entry will be there. When I say sufficient alkaline bicarbonates are not secreted, so I will not allow further entry of food bolus into the duodenum. The sphincter will remain closed. So what will happen? They will be stored in the stomach and produce hyperacidity. There will be distension of the stomach. So you will feel uneasy. You will feel nauseated. Okay, there will be vomiting, and with this regurgitation, what will be there? The food bolus will be entering the esophagus. And it may come out in the mouth. You will find, oh, so acidic it is. So it may cause duodenal, no, esophageal, ulceration in esophagus. Ulceration in esophagus. Okay, so it may produce, if it happens repeatedly, what will happen? Hiatus hernia. Actually, there is you have seen Trump, Dhak. Trump. So what is there on skin like thing? Say for what? Making percussions. Okay. So this type of thing is there separating. Here it is diaphragm. So there is a hole. Through this hole, esophagus enters. And this is the stomach. Now what happens? So part of the stomach, part of the esophagus here becomes inflated. So it is like this. A rudimentary stomach type. Okay. So this is another complication. This is called hiatus herniation. Herniation. Okay, this will give a lot of pain and acidity, burning sensation always. So these are the complications, hiatus herniation. All because of irregularity in our food intake, dietary intake. We are not following the natural phenomena. What is disease? What is disease? Disease is, it happens when you go against nature. When you go against nature, law of nature, then you are going to suffer diseases. So you have to follow. If you want to remain healthy, if you want to live a longer life, you have to follow the nature. What is happening? Why your foot, the base of the food, it is very thick. No? Why? It has been given. Why it is made thick?
Protects us from the harsh.
We will be walking, no? We will be collecting food. We will be collecting food. So for that purpose, we have to walk on the field. Okay? And we have to climb to collect food and to protect. If somebody attacks me, I have to run away. So it should be thick, and so that it can take the resist the frictions. So what we are doing, we are not using this. Always we are wearing shoes, and nowadays we don't walk. Whenever car will be going there, car is needed. A scooty, bike is needed. So we are not using it. As a result, you are not properly using the organs that you have been given. There is inactivity, and inactivity will create so many diseases, lifestyles. So, proteal action or proteolytic enzymes we learned. Okay. Enterokinase actions we already done. So, enterokinase the activation of trypsinogen into trypsin. Okay, this part is done. Digestion is done. Digestion of protein is done. Digestion of protein, when the protein will be entering, half-digested protein or partially digested, will be entering into the intestine. Pancreatic juice also will be coming. So there are trypsinogen is there, chymotrypsinogen there, they will be activated. They will show their activity. They are also endopeptidases. They are also having carboxypeptidases. They are also having gelatinase. They are also having elastase. So mutton, chicken, they will be having gelatin, they will be having elastin. So that has to be digested. So this will be digested by elastase, gelatinase, okay, chymotrypsin, trypsin, by this enzyme protein will be digested. Now, what is left? Aminopeptide, now peptides, small peptides, dipeptides, tripeptides. So, intestine, you have some enzymes, enzymes, with this you perform. the rest of the thing. So they will find, lot of chota peptides, small peptides, tripeptides, dipeptides. So they are having these enzymes. With these enzymes, they will complete the digestion of protein ultimately into amino acids. In this way, after full completion, we will get a lot of amino acids. Out of these amino acids, but remember, whatever the protein you are taking through food, is it possible to digest all the proteins? No. Some part will remain undigested. So protein is classified: simple protein, conjugated protein, and derived protein. We know what are the conjugated protein, and there are simple proteins. There are times different, we classify albumin, globulin. These are the simple proteins. Okay, histones, some proteins, scleroprotein. So we cannot digest them. Can you digest it? Though you eat it. Sometimes you find, suppose you will be eating nails. You cannot digest. So these are undigested protein. Even some plant proteins also remain undigested. Some animal proteins also remain undigested, so they pass through. Not that. If all the proteins had been digested, on examination of the stool, we calculate whether protein, how much protein is not digested, undigested. We calculate the amount of nitrogen present in the stool and amount of nitrogen the person ate. So if I subtract this amount from this amount, protein means nitrogen. These are only nitrogenous compound. So presence of nitrogen here is actually protein, undigested protein. So if I subtract this amount from the protein ingested, the balance is the amount of protein I digested. It has been absorbed into the blood. We have classified protein into various groups. First class protein, second class protein. What are first class protein? First class proteins are those proteins having all the essential amino acids. Having all the essential amino acids and in good quantities. And they are able to supply all the necessary essential amino acids, required amino acids in my body. So then this type of proteins are called first class protein. Example of the first class protein is egg, milk, and flesh.
They are just foods. They are foods.
These foods are having protein. These proteins are present in this source. Soybean. Is it a first class protein? No. Why? All the essential amino acids are not there. Or even it may be present but not sufficient. Similarly, for vegetable source, can you say the plant source of, rich source of protein? Beans. From bean we get dal. Dal. So they are actually good source of protein, but they are not first class. So those who are vegetarians, if they think I will survive nicely only eating the vegetables, not possible unless you take the milk along with. So you have to take milk to first. Then you will be getting all the essential amino acids. Okay? So you will not be having any deficiency. You will not suffer. Other amino acids I can prepare. My body can prepare. We are ready. We have the machinery. But essential, we cannot. That's why it's essential. You cannot prepare it, make it. That's why it is essential. Okay. Next, let's see. So this way, protein digestion is completed. In our next class, we will be studying how these are absorbed, how these amino acids are absorbed. Okay. So then we will discuss about the amylolytic enzymes, how carbohydrates are digested. So digestion starts here by the salivary amylase, and it is 1,4-glycosidase. So it will split the starch, glycogen present in meat, liver, or in muscle. Or in muscle, you are eating, we eat also muscles. So these will be having glycogen. So these glycogen and starch, these are polysaccharides. So these polysaccharides are digested by the salivary amylase. And what they produce? In my class I have shown, say dextrine, alpha limit dextrines, a lot of these, because these dextrines are not, a lot of branching bonds are there, means 1,6-glycosidic bonds are there. So because there are a lot of presence of lot of 1,6, so they cannot further digest the dextrins. Maltotriose, it is also having 1,6-glycosidic bond, so it cannot digest this. So in this way, wherever there is 1,6-glycosidic linkage, they cannot break it. They cannot even break the terminal glycosidic bonds. In between, the bonds in between they can break. So this way, ultimately, what we get by the action of ptyalin, their action will be here, and the food when will be coming to stomach, then it will, there also it will continue its action, but for a while only, because by this time acidity will be increasing, pH will be falling, so in this pH and acidity it cannot act. So action of this is over. And in stomach, are there in digestion of carbohydrate? Yes. Only one thing can be digested, means hydrolyzed. It is what? Sugar. And for this, there is no enzyme actually. There is hydrochloric acid. In your biochemistry class, you will find, what is this? Sucrose, when you perform Benedict test, you will find this is no result, because it is non-reducing. Okay, now if you hydrolyze it with hydrochloric acid, this bond will be broken. Now if you perform the Benedict test, now it will show the red color, means reduction, means this compound, means that has been split into glucose and fructose. So my stomach is also having hydrochloric acid. So with this hydrochloric acid, we can digest some part of sucrose into glucose and fructose. Is that clear? That much is the only digestion of carbohydrate in stomach. So thereafter, all the through food bolus, so this will enter into the intestine. Now it will face two source. One is succus, entericus. These are the enzymes, amylolytic enzyme. And from pancreas, it will also get pancreatic amylase. And other than this, maltase is also there. So they will come into action. Two sources. One source is this, another source is pancreatic juice. So pancreatic juice, see, there is no 1,4-glycosidase here. So all the rest part of starch digestion will be completed by pancreatic amylase, by pancreatic. So starch digestion or glycogen digestion will be completed by pancreatic amylase. And the products they will produce, they will be digested by these enzymes. Sucrose will be digested by sucrase, and what will be the product? Glucose and fructose. Maltase, it will act on maltose, so it will act on this. So 2 glucose unit will be formed. Lactase, they will break lactose into glucose and galactose. And there will be dextrinase, lot of 1,6-glycosidic linkages, so it will break the 1,6-glycosidic branching. So this is one chain, it has her 4th carbon has produced one link with this, then from this 1,4,1,4,1,4 link. So if I can break this branching point, I can break this. Now this can be digested by 1,4-glycosidase, means pancreatic amylase. Okay, so I have to first break this branching point, cut this branch. This is done by dextrinase.
So why do you need such enzyme if the carbohydrates can be digested by HCl only?
No. It cannot break other, it can hydrolyze other. These are weak. And the enzymes can do it, make it very fast. Dextrinase, trehalose I told already in the class. And the lipolytic enzymes here, some lipolytic enzymes are present. Salivary lipase, its action is very poor and it acts on only emulsified fat. And stomach is also having gastric lipase. It also acts on butter fat, milk fat that can act, not complete. So thereafter, the food when enters into the intestinal lumen, there it faces 2 lipases. And esterase, pancreatic esterase, pancreatic cholesterol esterase, phospholipase A2, phospholipase A1, different type of phospholipase. So they will complete the digestion of lipids. And if anything left behind, that will be digested by this lipase. So in this way, fat digestion may also be completed. Okay, so you came to know how proteins are digested, how carbohydrate is digested, how lipids are digested. But lipid digestion is not so simple because the oil droplets are big and they cannot bind with their enzymes. So what has to be done? Their size is big. And it is the enzyme and it is the binding site. The oil droplet is big like hathi, elephant, and it is the binding site. Can it sit there? No. What has to be done? It should be emulsified. Its surface tension, if we reduce the surface tension of the fat droplet, it will be emulsified. Fine droplets will be put. Now this droplet can bind with this. The size is okay size. चलो अभी तुमको तुम्हारे लिए है हथौड़ा मेरा पास. So this will be digested. And to emulsify this, we need help of bile. What is there in bile? Bile salts. They will reduce the surface tension. As a result of this, we will get emulsion. Then only possible. Now, in absence of liver function, proper liver function, will you be able to digest? No. Will you be able to, if somebody, for you, if somebody have digested this level, can you absorb it? No. Because for absorption, you have to form the micelles. Otherwise, this will not be absorbed into the lacteals. I have already discussed. Chylomicrons, formation of chylomicrons is needed. Bile salts are needed. That's why bile salts are important. So, that's why through bile, it is secreted in the intestinal lumen. Do you know that these bile salts are again reabsorbed because these are very important. Again reabsorbed by the portal system and will be sent back to the liver. Again these bile salts will be liberated through bile. This is the circulation of. Question comes. Hepatoportal circulation of bile salts. What is this? Explain it. In this way, these are recirculated, and it is recirculation of. I have liberated 100 bile salts, and absorption will not be 190. So always there will be some loss. So for that we will be prepared, like liver will be preparing this. Understood? So this has to be in your mind always. Okay? So function of mucus, you know it very well. And intrinsic factor, absorption of vitamin B12 and defense. It provides defense to your protein. Now it is told what is this function. It will provide immunity. It will prevent inflammation of intestinal wall by the foreign organism. Okay, so now we could understand the functions of intestine, the functions of succus and enteric. Is that clear now? Okay, now there is control. How these are controlled? Regulated. Again, by two processes. One is neural control, another is humoral control, means controlled by hormones. Defensins are secreted by Paneth cells. Right? Paneth cells. I will place short questions and MCQ. Or we will ask, name the cells that secrete defensin. And function of defensin is these. Which one is correct? Okay. This is nice thing, no? We have some beneficial bacteria in our intestine, no? So it will make balance. Other bacteria, I will not allow. Bacteria, beneficial, you keep. So I will make balance between the two. In absence of defensin, this will be imbalanced. This mechanism of bicarbonate formation is same like the intercalated discs. They are producing bicarbonate ions, and there are two pumps and one exchanger is working there. Here also 2 pumps, 1 exchanger is there. Same procedure. With the same procedure, the cells, these cells of intestinal cells, they can also produce bicarbonate alkaline. They make the solution alkaline, the secretion alkaline by this procedure, regulation. Secretion of succus and enteric regulated by both nervous and humoral mechanism. Nervous mechanism means by parasympathetic and sympathetic through vagus. When vagus nerve is stimulated, there will be increased production of enteric juice. Intestinal juice, succus and entericus. But sympathetic stimulation, just opposite. It will reduce the secretion. And mostly, nervous control they do through local reflexes. Local reflexes means when food will be entering into the intestine, so they will create lateral pressure, distension. So there will be epithelial cells, they will be pushed.
Yes, sir.
There are some receptors. It will sense this pushing. So now they will sense, they will be stimulated. It immediately send impulses to the Meissner's plexus, not to hair center. There are, I'll discuss in detail, there is one nervous system in the intestine, in the GI tract, that is called enteric nervous system. So they have got, these are local, no Usually, higher centers don't interfere with their work. But if necessary, they can interfere. Okay, so how they work? When there is change in acidity, more acidity, this reflex will be stimulated. When there is any distension, they will be stimulated. So, to minimize this distension, to minimize this acidity, what they do reflexly, they will secrete more alkali juice and they will produce more mucin so that this food, when it is pushed, these cannot damage the wall because the food surface of the food will be slippery. So in that way, the local reflexes prevents this thing, helps digestion. Understood? So, and this is the neural mechanism, and lastly, regulation of secretions by these local stimuli, distension, chemical irritation by hydrochloric acid, presence of hydrochloric acid, and role of VIP. VIP means? VIP means? Very important person. Yes. And in this case, VIP means vasoactive intestinal peptide. Okay. So they will secrete vasoactive VIP. Meaning it will affect the blood vessels. Vasoactive means it will always, our blood vessels, there is smooth muscles, no? So they have some tone. So it remains always in some contracted form. Okay, now I can stimulate this. Okay, so what will happen? This tonicity will be withdrawn. So this will be dilated. So there will be more passage of blood. So if in the gland more passage of blood is there, they will produce more fluid. So in this way, VIP can increase the secretion. Can you follow? Could you follow? What did I say? So vasoactive peptides, they will cause dilatation of the blood vessels, means increased flow of blood to the tissues there which are producing enzymes or these secretions. So this will be increased, alkalinity will be increased, production of enzyme will be increased. Brunner's gland, I have shown you the Brunner's gland. Have you seen that in class? In the submucous layer, round, they produce mucus, mainly mucus. Yes. These are all these gastrin, CCK, PJ. CCK means cholecystokinin. PJ means pancreozymin. Previously we thought these are separate. One will cause secretion of pancreatic juice, and other is contraction of the gallbladder. And later it came to know both are actually same, but they are having both the actions. It can cause contraction of the gallbladder as well as secretion of pancreatic juice. Okay, another is secreting VIP, GIP. VIP to समझ में आ गया, vasoactive intestinal peptide. And now what is GIP? No. Previously it was thought it is gastrin inhibitory peptide. Gastrin inhibitory peptide. Now its name has been changed, but abbreviation is same. What is the name? Glucagon-like substance. Glucagon-like. That's why G is there. Okay. Actually, it is at the same time. That's why name has been changed. At the same time, it is glucagon or glucose. Actually, it stimulates insulin secretion also. Glucose-dependent insulinotropic hormone, peptide. Its secretion starts in the intestine when food enters into the stomach. It assumes that lot of glucose will be forming. So, Pancreas, be ready with secretion of insulin. Because lot of glucose will be entering into the blood. So you have to manage it. You have to bring down, there will be increase in glucose concentration in blood. So you have to bring it down to the normal. That's why insulin secretion is increased prior to that. So that's why name has been given to like that. What is the name?
Glucose dependent.
Glucose, you keep this. This is the latest name, not because it is having also role in insulin secretion. That's why name has been changed. Glucose, when intestine, glucose not yet absorbed. Before that, it sensed a lot of glucose will be entering into the blood. It sensed a lot of food is coming. Already some glucose I have got. So this glucose has stimulated me, and now I could sense a lot of glucose will be formed, and blood levels of glucose will be increased. There will be hyperglycemia. So it has to be by any means. You are normal individual, your blood sugar level should not be beyond 180 mg/%. So if beforehand insulin is not ready, then it may shoot above 180 sugar level, and it may cause damage. We are concerned, diabetic patients are concerned with increased level of sugar. Why? Because this increase, you will find, you are going to be doctor, so you will remember this. Every food, every carbohydrate has one glycemic index. You might have heard this word, glycemic index. Don't advise any carbohydrate to a diabetic patient whose glycemic index is high. Means those carbohydrates which are digested rapidly, absorbed rapidly, their glycemic index is high. They are capable of increasing blood sugar level very fast. Rasgulla. Sweet things. Their glycemic index is very high. That's why it is this potato, banana. So their glycemic index is high. That's why we will be advising carbohydrates whose glycemic index is very low. Not refined maida, flour.
That is high glycemic index.
High glycemic index. That's why it is not advised for diabetic patient. We recommend atta. Oats. Their glycemic index is low. So you got the idea what is glycemic index? You remember this. Saras told first.
Nice, good. Always maintain the pressure even if you have to breathe again. Okay, nice. Okay, whenever it goes beyond 101, immediately you have to seal that red cap. Okay, again try. No, it is not. It has not gone beyond 101. Again, drop it in the fluid first. Yeah, now draw. Bus. Close it, seal it. You know how to seal, right?
Yeah, we have to hold this part, right?
You have to seal this one. You have to break this rubber tube. See, it will, else it will drop. No, you are very slow. You have to be fast, right? You have to be very fast. See, it has gone beyond. I think it has dropped below 101. Yes. Seal. Yeah. Seal first. Okay. Bring it up. See? No, it's again. Now it's there. Okay. Now discard till 101. How do you discard what I have told you? You have to use this one. You can touch it there. Wait. You have to see. नहीं वो बहुत नीचे चला गया. Again, draw again. See. Use this and use it like that. इतना दौड़ तो. See, you have to break this one. You can't let any fluid come out of this pipette into the diluting fluid. See, there is basically a bit of blood is there already. Okay, so it's okay. It's your first time, so do it again. Okay. Okay. Don't let any air enter this. Yeah, draw, no problem, I'm seeing. Again. बस block. ये अभी ठीक है. Now mix it. Keep it horizontal and mix it. Okay. For how many minutes you have to mix it? In between palms, horizontally, right? Mix. You can let the rubber tube hang from your palm. Let it hang. No, it will not come. Just mix it for 2 minutes, right? Then who is going to do the charging? Achha, both of you have to do the charging, right? Tissue paper and ulta koi tissue. Achha, turn on the let me clean it. It's okay if the time passes beyond 2 minutes. There is no harm in it, but it should be minimum of 2 minutes so that it gets evenly mixed with the diluting fluid. Take the pipette.
We have to drop.
Yeah, of course. You have to drop. No, don't need to blow it. It will come automatically. If it doesn't come, then only you blow. See, it will come. Okay. Keep it in a 45-degree only, so that you can directly bring it to the new birth chamber. Okay. Okay, it's okay. You have dropped 2 drops, right? Now you can charge. No problem.
So you don't need to blow now.
No, it will come now automatically. But make an angle of 45-degree, right? Wait. Do 45-degree and only touch the drop at the edge of the cover slip. Okay, not over, only at the edge. You can see. You have seen, right?
Yes.
That's the charging. Do RK korbe. It's very good. RK korbe.
Charge the other side.
See, do it very carefully, right? Let the drop come and touch. After the drop comes, wait for the drop. Make it a 45-degree angle. Make it in a 45-degree angle. Okay.
Yes.
नहीं हुआ है. You should let the drop comes first. If it doesn't come, try to give it a force by your thumb. It will come first. See, it's coming. No, I think it got blocked by that drop. Always try to make it in the edge. It will not get. Okay, so it got blocked basically. So you have seen the charging, right? Okay. Yeah, let it be there, no problem. Okay. No, there is already EDTA sample in there, anticoagulant. This one.
So what do we call this tube?
This one. This is actually blood container only. There are other areas. Pathologists use this, and there are various colors are there. This color coding is basically used for what type of anticoagulant is there, and when we take the blood, how many times we have to mix it. For purple color, we have to mix it till 8 times, right? 1, 2, 3, 4, 5, 6, 7, 8, right? 8 times. Take.
You can do it.
Okay.
So দেখা যাচ্ছে না কিছু
দেখা যাচ্ছে
ক্যাপিলারি
she is seeing. Draw. No, you have drawn here basically. Again, blow it out. Okay. Wait. Now draw. Bas. See what you have to do. It's see the reading first vertically. Okay, what is the reading?
1
More than 0.5, right? Now what you have to do?
I'll soak it with.
Okay, you have to soak it also, and also have to wipe the outer part so that the blood doesn't get mixed with the diluting fluid. Okay, just touch very gently, okay, and touch and remove. It will come out very fast. Bus. It's very good. Next, who is going to do the? Okay, you can do.
ক্যাপিলারি অ্যাকশন মানে
एक मिनट मेरे से कोई बात नहीं शायद मैं बता दूं उतना ही एक और वेरी गुड वेरी गुड बस हो गया अरे आपको 45 डिग्री में स्टार्ट करना बहुत सही है।